Good afternoon, ladies and gentlemen. I'm Navindra here. I'm moderating the session today, which is we have got a esteemed speaker from Singapore with the title of Meliodosis, Burkholderia pseudomalai, navigating identification pitfall and AMR, which is antimicrobial resistance. Before we start, can I just briefly introduce him? The speaker is Professor Timothy Mark Sebastian Barkham. He's a senior consultant in the Department of Laboratory Medicine, Tan Tock Seng Hospital, Singapore. And he has been adjunct associate professor in the Department of Medical Microbiology and Immunology, Yong Lu Lin School of Medicine, NUS, Singapore. He obtained his MBBS in 1988 from the St. Thomas Hospital, London, UK, and then followed by the Masters and Fellow of Royal College of Pathologists from the UK. His expertise involved clinical and laboratory leadership in various areas. He also has led outbreak responses, such as he played the frontline role in developing and deploying rapid diagnostic pipelines ahead of the or during the major regional outbreaks, including the SARS in 2003, H1N1 influenza, chikungunya, dengue, and COVID-19. He has got numerous numbers of publications in index journals. I wouldn't want to bore you with his extended CV. Without further ado, in the interest of time, I pass the floor to you, Prof. Timothy.
Thank you very much. So thank you for inviting me. I saw this quote in a journal that I was reviewing, and it said that with early diagnosis, best antibiotics, and the best ICU treatment, healthy people should not die from melioidosis. I thought it was quite a brave statement. But if you look at it closely, you realize the problems. Many of our patients do not have early diagnosis. Many of them are starting.
Prof, can I stop you? I can't hear you.
Places. And of course, most people with these numbers. So it sounds very but unfortunately, it's a document published a few years ago. It may kill more people each year than dengue. Well, that made me stop. I hope it makes you stop and think. It accounts for about 20% of community-acquired septicemia in Northern Australia and Northeast Thailand. And it's the most common cause of severe community-associated pneumonia in Northern Australia. I think these items here make it sound very important. And I came across a poster at a meeting on bacteremia in Nakhon Phanom, Thailand. E. coli is number one, we're not surprised by that, but number two is Burkholderia pseudomalai. It's even more than Staphylococcus aureus. It's double the Staph aureus, look, 73 and 134. That's amazing. So, of course, the issue is, we agree it's important, but can we do better? It's clinically difficult to differentiate from other infections. And that's important, guys, because that means that the doctors diagnosing these patients don't give the correct therapy because they don't realize it's melioidosis. And it often progresses rapidly to septic shock, multi-organ failure, and death. So often by the time we've grown an organism, actually the patient's already dead. We can ring up with a positive blood culture saying there's a gram-negative rod here and the patient's already dead. The gold standard for diagnosis is culture, but of course it's a little bit slow. And of course sensitivity is not very good. It's only about 60%. So you can't believe the negatives. The negative predictive value is not very high. And of course, pseudomalai is resistant to many empirical regimens. We do need particular antibiotics to treat this organism well. So we want to do better. We want to get the faster diagnosis. Earlier therapy with appropriate antibiotics can result from a faster diagnosis And hopefully this should decrease the mortality. And our benchmark, the thing we look at, is the data from Australia. Their mortality dropped from 30% to 6%. That sounds fantastic. So the main aim, I could stop the whole talk here and just say the main aim is awareness. You've got to have people aware of this organism, what it looks like, and that it might be on the plates they're looking at in the laboratory. The sort of three areas we're thinking about are the diagnostic problems, how to identify it quickly, how to be suspicious of it quickly, because if we're suspicious of it, we can be safer, because we're worried about safety in our laboratories. And of course, there's the antimicrobial susceptibility testing, the problems with emergence of resistance during therapy. We want to get all these things right so we can help our patients and our staff. So I'll talk a little bit about the history of the organism of disease, because if you don't know about the history, the rest of it's a little bit boring. The history and understanding the disease make it so much more interesting. We'll talk through culture, susceptibility testing, identification, a little bit on serology and nucleic acid tests, and safety. And I will end with a slide asking you guys what you do in Malaysia. So before we talk about melioidosis, I think we should actually talk about Burkholderia mallei, because our organism is pseudomallei, but it makes you ask the question, what is mallei? So Burkholderia mallei causes glanders, also called mallei or farsi. And this is an animal disease, and the primary host are solipeds. Solipeds are animals with a single hoof, like horses, mules, and donkeys. And this organism is found in soil and water. Sorry, it is not found in soil and water. So what's interesting here, if you look over here on the right, mallei apparently is a clone derived from pseudomallei, and it evolved through gene loss and selection, became adapted to these solipeds so that it can no longer survive in the environment. So mallei actually has very different epidemiology compared with pseudomallei. Soil and water are not reservoirs for Burkholderia mallei. Now, around the world, animal infections are effectively eradicated in many of these places listed here, but it's still endemic in other parts of the world. So again, at the end, it'd be lovely to hear from you if, do you actually see mallei? Maybe, I don't know if any of you work in the animal industry, but I've never seen mallei. So let's just move on to melioidosis now. It's fun to read early reports. So I've listed some of them down here. The earliest one there being in Whitmore and Krishnaswamy in 1912, when they were doing necropsies on vagrants in Rangoon in Myanmar, and they described the disease as fulminant and rapidly fatal. Or they also described cases where people had chronic problems over years. So they did a very good job, actually, even back then, without all the technology we have. They also said it can resemble tuberculosis, and I looked it up, and I understand in Malaysia reported around 26,000 TB cases a year or two ago. Why do I put that there? Is because it's inevitable that we confuse the diseases in our patients, because TB can also be very difficult to diagnose. Again, it's awareness, what we're looking for. So where did the word melioidosis come from? Actually, it was proposed by staff at AMR in KL. They proposed it because the word melis means similar to glanders. I've just told you what glanders is. Melis means that, so they used the word melis to propose melioidosis. And where does the word Burkholderia pseudomallei come from? Well, it's pseudomallei because it's like glanders, but Burkholder comes from the name of a chap who described Burkholderia cepacia, which he discovered when he was investigating the cause of an outbreak of onion rot, which I always think is quite funny. So these early reports described a mortality as high as 98%. So that's very, very high. They also said there's a disease of Southeast Asia, but their excuse for publishing it in a Western journal was that there are returning travelers going back to all parts of the world that may take the disease with them. So people should be aware. So again, it's awareness. Otherwise, you won't make the diagnosis. Now, at that time, they didn't know what the source was. They associated it with morphine injectors, and it was called morphine injector septicemia. A lot of these vagrants in Rangoon were morphine users. They did say there was no person-to-person cases, and they had found it in lab animals and in rat feces, and this led to this publication saying a disease of rodents communicable to man. So they didn't really know everything that we know now. And of course, now we can do a lot better. Mortality, according to the Australians, they can bring it down to about 6%. We know it's not just in Southeast Asia, it's all over the world in tropical areas. And we know where the organism comes from. It's from the environment. So a quick clinical overview: about half the people come in with pneumonia, half are bacteremic, and 20% have septic shock. And the mortality in septic shock is about 60%. So it's a severe disease. Interestingly, skin infection seems particularly common in Australia without sepsis. And parotid abscesses and other infections around the oropharynx seem to be more common in children in Southeast Asia. So we do get these regional differences. And there's another one here: genitourinary sepsis with prostatic abscess, apparently particularly common in Australian males. But I think that's because the Australians have the resources to scan their patients. So it may be an artifact of who has the resources and who actually bothers scanning to look for the prostatic abscesses. But the organism can be found in any part of the body. And that, of course, is the problem. It may grow on any bench in your laboratory. So from the safety perspective, from the awareness perspective, from the perspective of you looking out for your safety and for a quick diagnosis, we need awareness amongst all the staff in our labs. So just one or two more things on the epidemiology. We've mentioned the soil and the water in the tropics. People may acquire it from percutaneous exposure, inhalation, aspiration, or ingestion. But it's not just wind and rain. It's also unchlorinated water. It might be in your pipes. Interestingly, the seropositivity reached up to 50% in northeast Thailand. So as you read the data, you start to think, wow, it's all around us. And the message, of course, guys, for you now is don't get diabetes. Don't get other illnesses to protect you from melioidosis and other things. Interestingly, there's a paper talking, this is a Nature Review paper just 2 years ago, talking about latency, saying it's actually quite uncommon. And most of us, when we talk about melioidosis, we tend to think that latency is common, but apparently it's not. So they reviewed almost 1,200 cases, and only about 3% of them possibly arose. Okay, still uncertain there. Possibly arose from latent infection. A lot of the cases were chronic cases, which have been misdiagnosed, and therefore people subsequently described them as latent, but the authors thought that was not correct. Interestingly, there are some cases described in America, which they felt the patients had latent with cases, other cases from around the southern United States. So they think it was actually locally acquired in the US, and that the long latency story was not correct. Oops, sorry. So again, awareness drives diagnosis. So we see that the epidemiology that we began talking about this in Myanmar and Malaysia and Australia, but now we find actually it's all over the Pacific and Indian Ocean islands, in Africa, Central and South America, the Caribbean. And genomic analyses suggest the organism spread from Australia to Southeast Asia during the Ice Age, then to Madagascar, Africa, and then on to the USA. But Malaysia, well, you are an endemic hotspot, and it's widely distributed. So have a look at some of these rates. And notice the rates in Kedah are approximate to those really high rates in northeast Thailand and the top end of Australia. If the rates are not that high in your part of Malaysia, it might be because it's been underdiagnosed. Of course, you don't know. It's also been found in many different animals. So the lab aspects, everybody wants us to diagnose this. They want to catch it quickly for the patient's sake and for our sake, so we can handle it safely. And people will start describing this organism like this. They say it's a motile oxidase positive gram-negative rod. But you don't know if it's motile or oxidase positive or gram-negative when you open the plate and look to see what's growing. So it's almost too late. And they describe bipolar staining and the safety pin appearance, which I can show you here from a picture. But most of our staff would not predict milii from this. We had a bacteremic case at the weekend, just gone this last weekend, and the staff did not alert anybody to a safety pin appearance. Retrospectively, we go back and go, oh yeah, we can see one or two safety pins. But I don't think it's a terribly useful tool, because I don't think it's that reliable. So my question to you is, what of these things really help you? Because actually what you begin with usually is a plate and you find some more bacteria growing on them. What you really need is awareness and an idea of what the colony looks like. Okay, because you may do a gram stain, but by then you've already manipulated it on the open bench. So it's the colony awareness which I think is perhaps most important. And actually nowadays, we don't even do a gram stain. We dive straight in with a moldy. So this safety pin appearance isn't something that's going to help us very much. It could do on blood cultures, but we don't rely on it. All our blood culture bottles are subcultured in the biosafety cabinet. So the gram stain, the slides are prepared and dried on a hot plate in the safety cabinet. Of course, we then take the dry slides out and stain them in the open, but the organisms don't jump off the slides, so I think that's perfectly safe. So when you do open the plate and have a look at it, well, the organism, when it does grow, grows pretty well on routine media, blood agar, chocolate agar, McConkey agar. So I'm just checking for messages from you guys to make sure I'm okay. Earlier colonies can look very different from older colonies. When you see a colony like this, then it's unlikely to be many other things. Once you see these wrinkled colonies, you wonder to yourself, could it be Pseudomonas stutzeri? Because that also makes nice wrinkled colonies. But usually it's not. We don't see stutzeri very often. It's nearly always Pseudomallii. The earlier colonies are not so specific. So traditionally people talk about it as dry and wrinkled and having a characteristic metallic sheen. I'm not actually so good at appreciating the metallic sheen myself, but that's what all the books say. What we do see, that is also written in the books, is you can have a mixture of mucoid and non-mucoid colonies in the same sample. So here's some lovely pictures I borrowed from this article showing blood agar and TSA, these non-mucoid ones on the 2 agars, mucoid on the 2, and then a mixture on the blood and on the TSA. And these are the same organisms, genomically they're identical, it's just different expression of different LPS in their cell wall. What do we do about trying to find a melioid from non-sterile sites that may be mixed or overgrown? Well, people use selective media, but actually we don't, not at my hospital. And I'm not sure anybody else in Singapore uses it all the time. We don't see enough of the disease to warrant doing it all the time. You would have to have somebody ring up and say, hey, can you dig the power guys use. We should all be familiar with selective media in the literature, the Ashdown agar and Ashdown broth. They use gentamicin or colistin, crystal violet as selective media, because traditionally most Pseudomallii is supposed to be resistant to these drugs. And traditionally, a lot of the other contaminating flora is susceptible. Although nowadays, with more microbial resistance in our doctor-exposed patients, we're seeing more and more resistant organisms that grow on these agars. You can also use a C. sepacious selective agar. Again, this relies on gentamicin as well as other antibiotics. And here's a media that I read up about that actually was described by a group in Malaysia, Francis medium, that uses a higher gentamicin concentration, 8, compared with the typical Ashdown agar. Now, of course, I've got to mention that Sarawak is special. The Burkholderia Pseudomallii in Sarawak has been shown that about 86% of all their isolates are susceptible to gentamicin. So this actually is rare elsewhere. It's restricted to particular sequence types. This is sequence types according to MLST or single locus variants of these sequence types. And it's due to a mutation of a component of an efflux pump. So the interesting thing then is, if you are using selective media, you really do need to know whether the isolates in your locality are gentamicin susceptible or resistant. So I would wonder, well, what do they use in Sarawak? Perhaps you guys can tell me at the end of the talk. So I come back to early identification, and really it comes down to awareness, which our older staff are better at being aware, and they consider stuff. But our younger staff are not, and they are perhaps more likely to make an error or to handle something on the open bench, which we don't want them to do. Because we moldy everything, the rest of this slide is not so relevant to us because we moldy things first. We moldy them before we have a gram stain. We moldy them before we have an oxidase result. We moldy them before we have a susceptibility result, because Pseudomallei is typically resistant to gentamicin and colistin and susceptible to amoxicillin clavulanic. So for us, because we have moldy, the problems other labs may have are not problems for us. But I understand from the literature that the latex agglutination is widely used. So this test uses a monoclonal antibody against an exopolysaccharide. But I'm not aware that anybody uses this in Singapore. So let's talk about the moldy. Awareness in moldy. It's much faster than the biochemical methods. Of course, it literally can take a few minutes. But our database does not include Pseudomallei in the database. So it reports Pseudomallei as thailandensis, Burkholderia thailandensis. So what we do is we then take our the output from the moldy and send it to the public health lab, and they have a special database called an SR, a security relevant database, and that reports our organisms that we call thailandensis. They report them as Pseudomallei stroke Mallei, because the moldy cannot distinguish between Pseudomallei and Mallei. So for us, it's straightforward. As soon as we see thailandensis, we know it's going to be Pseudomallei. So this is the report that we get sent. So this is one from last weekend, 17th of August. So a score above 2 is taken as a pretty reliable score to species level. So we have Mallei came out top, Pseudomallei, Mallei, Pseudomallei, Mallei, all the way down. And then suddenly there's a Burkholderia thailandensis. Okay, but this is Pseudomallei and we report it as such. So what is thailandensis? And we'll stop for a moment and just make sure we know what that is. It's a soil organism, like Burkholderia Pseudomallei, but it's 1,000 times less virulent. And it's not a BSL-3 organism. It's arabinose positive. So in the olden days when we used API 20NE, we would always look at the arabinose very carefully. But the trouble is, Pseudomallei, about 25% of them are also arabinose positive. So the arabinose alone does not tell you the whole story. And of course, reading these biochemicals is a little bit subjective. My understanding is in the literature, there are only about 6 human cases of thailandensis. And some of these are uncertain anyway. So the pretest probability that you have a human infection with thailandensis is close to 0.00 something. Okay? So if you get an idea as thailandensis from a human sample, it's probably incorrect and it's probably Pseudomallei. Thing about the commonly used ones like API 20NE is that, of course, the interpretation is subjective. And the most common incorrect idea is capacia complex. You can try to distinguish it by looking at the antibiotic susceptibilities, because capacia is normally resistant to co-amoxiclav, whereas Pseudomallei is normally susceptible. Capacia doesn't cause the wrinkling, so you keep the plates up for a few days and see whether it wrinkles. Of course, you can look at individual biochemicals like the ALO. If you have antigen detection available, which we do not, of course you can use that. We do have a PCR available, and in theory we could do sequencing if we needed to, but we don't because we have the moldy. There's also the VITEC system, and this has also got an interesting story to it, because again, if we look at Sarawak, the VITEC 2 has been reported to misidentify Pseudomallei from Sarawak in about 35% of cases, so 15 out of 43 from Sarawak were misidentified. But if you look at isolates from Sabah, there's only about 2 out of 25, that's 8%. And Darwin, Australia, they've looked at about 150, and only 3 of these were misidentified. So people analyzed it, and in this paper from just over 10 years ago, they explained that the biochemical profiles of misidentified isolates were actually very different for these 2 particular tests here, and they were down to different constituents in the cell wall of the organisms from Sarawak. And of course, they define these organisms as Pseudomallei based on PCR and latex agglutination. So it's an interesting philosophical point here that we should expect difficulties with other systems. In fact, with any system we bring in from another region in the world may not work so well with our local bacteria or viruses or whatever it is we're doing. Systems do need to have local validation. even in different parts of a country, especially when your country is so large, like Malaysia is. So this was a case where people weren't sure, was it Pseudomallei or Thailandensis? So this was in EID in 2017. They talked about a case from isolated human infection in China in 2013. So they claimed that the NE had misidentified the organism as Pseudomallei, but when they reinterpreted the results, they identified it as Thailandensis. And they made a particular note that it was Arabinose positive. But I'm hesitating here because I'm now feeling uncomfortable because I'm reading a story where in the same story they then said the VITEC2 identified it as Pseudomallei and 97% probability. So you're thinking, okay, which one is it? Which one is it? They did a 16S RNA ribosomal DNA sequencing and found 100% similarity with a sequence that belonged to Thailandensis. So they said that this was a case of Burkholderia thailandensis. However, if you look up how good is 16S ribosomal DNA sequencing for Burkholderia, if you look it up in the CLSI M18, it says 16S can be used for resolution to genus, but limited resolution to species. Okay, so it's a very useful document. You can look down this document and it'll tell you whether the authors think that 16S is a good method for different bacterial genera. So clearly they think the genus is okay, but not to species. So they actually say it does not distinguish Pseudomallei from Mallei. It says it can distinguish Pseudomallei from Capacia, but it doesn't specifically mention Thailandensis versus Pseudomallei. It just makes the comment limited resolution to species. So there was a rebuttal to this paper in EID that came from a group from Thailand and Laos who've had a lot of experience with Pseudomallei. And they said they thought this case report was incorrect and it was unlikely to be Thailandensis. They said the 16S is not reliable. The Arabinose in the NE is also not reliable. It's easy to misinterpret. And the authors from China had described virulence of the organism in mice. And the rebuttal argued that the virulence resembled Pseudomallei and didn't resemble Thailandensis at all. And the strain apparently contained virulence determinants, which actually are not normally found in Thailandensis. So of course, we can't be too sure who's correct, but the purpose of showing you this case. The other point is that we must interpret test results wherever we get them from, even from computers. We have to always ask ourselves, are we sure? Are we happy with this? Are we uncomfortable? So another example of difficulty making the diagnosis. So this happens to be a report from Hong Kong. They had a blood and sputum isolates from an elderly lady with bronchiectasis. She was acutely unwell with bacterial pneumonia, and they grew Burkholderia. And it looked just like Pseudomallei. It grew aerobically, not anaerobically. It was wrinkled. It grew at 42 degrees, but that's not specific. It was oxidase positive, motile, and it was resistant to polymyxin. The VITEC system, to their surprise, reported it as unidentified. Unidentified. So that was a bit of a surprise. It was Arabinose positive, so that was a little bit surprising. And the VITEC system, according to the literature, should be able to identify the majority of Pseudomallei strains, but this one called it identified. So they did an analyzer of the patient in both acute and convalescent sera, and this ELISA is meant to be quite sensitive, but this was negative. So that was a little bit surprising, so they turned to sequencing. But before we get the sequencing, let's look at this data on biochemical profiles. Percent of Burkholderia Pseudomallei showing positive results, and here is Arabinose. Arabinose, 25%. So you can't use the Arabinose positivity to say that the organism is a Thailandensis. It might still be Pseudomallei. So let's get back to the sequencing. So these were their results. They did the 16S, and they found no base differences between their patient's isolate and Pseudomallei, a particular Pseudomallei in GenBank. They found less than 1% difference between the patient's isolate and Thailandensis. They think the GROW-EL is a far more reliable place to sequence. So again, they found 0 to a few differences in the patient's isolate compared with other Pseudomallei strains, and a much larger difference, look, 2, almost 3% difference between their patient's strain and Thailandensis strains. So they concluded it was Pseudomallei. So I'm just putting those there to point out that there are problems. And this is an old but interesting and helpful publication. There was a diagnostic workshop about 10 years ago showing the performance of NE, Phoenix, and Vitek. And we see here for different parts of the world, some things in the 90s and 80s, but look, Australia here, 37% correct, 0% in this particular publication, 1981, and so on and so on. So again, this is awareness. Don't rely on one thing to give you the result. Make sure everything fits in. So they ended up, and this was meant to be a diagnostic workshop to make things easier for people in less well-resourced areas. So again, they start off with a gram-negative rod that Seok stays positive and not Pseudomonas aeruginosa. I think that's very reasonable. But of course, by the time you've got these results, then your patients or your laboratory staff have been manipulating it. Flat wrinkle colonies after 60 hours, after a few days, yep, that helps. Susceptibility testing, sure, that helps. If you have these tests, that's great, antigen detection, but we don't. So it then comes back to these biochemical identification. They then say below the dotted line, if you're in a more well-resourced area, you may be able to use Vitek or 16S or PCR. And of course, back in 2015, MALDI was pretty novel, so they didn't mention MALDI. So the situation in my lab is very different. We use MALDI. By and large, it's very easy for us to detect. And if the staff have a mixed plate, they can MALDI several different colony types. So the chances are they will pick it up. So interestingly, some people suggest using throat and rectal swabs. And there was a debate in the literature recently, 2026, and then there was a reply from an author. So basically, they're suggesting that routine throat and rectal swabs should be done in all suspect patients. But somebody else said, no, that's too substantial workload. We can't do that. There's a low overall positivity. Please don't do it. But then the people who work in areas where they're not so well resourced says, well, we don't have blood cultures. So actually, it may be reasonable for us. So if clinically it's suspected, it's a relatively easy, cheap, and non-invasive test to do. So please don't write articles telling everybody else not to do it, because we think it's useful. So they ended up concluding more common than in Northern Australia. So it was originally the Australian authors saying don't bother with these throat and rectal swabs. But people working in other parts of in parts of Southeast Asia are saying, actually, for us, it's useful. So they agree, yes, a targeted approach is preferable, and throat swabs perhaps should be prioritized in suspected melioid, in children and patients unable to produce sputum, in patients like Southeast Asia with more head and neck involvement. So you may choose. I don't think we would choose to do that in Singapore. So let's come back to lab detection. Well, culture's slow with a low sensitivity. So there are these other methods, direct staining, serology, and nucleic acid tests. So I've never seen this used, but, and I note that this is from 2005, a report saying rapid immunofluorescence microscopy done directly on the clinical samples on sputum had excellent specificity, but again, pretty low sensitivity. I've never seen it used. I wonder whether you use that. Real-time PCR apparently is very good. So the one that performs well is this one that detects the type 3 secretion system, very good sensitivity and specificity on various sample types, but not quite so good on blood. So in patients with septic shock, it might approach 70-odd%, but patients without septic shock, there's a lower load. It's not nearly as sensitive. They also mentioned that you can get false positives, and they're not too sure how to explain them. They weren't too sure whether these apparent false positives were real or not. And of course, they said it might be due to contamination or cross-reactivity, but it might also be detection in patients who were misdiagnosed. Maybe they did have melioid. Okay, but in the evaluation, the patients have been classified as not melioid. Or maybe they just had asymptomatic carriage of the organism. This is all possible. And they made the point that most of the false, if not all the false positives, came from swabs from mucosal surfaces. So things such as carriage could be true. So they ended up recommending the PCR only be used, for these sample types, not for throat or rectal swabs. So this is an interesting paper. Again, we don't use these things, but it's interesting for me to read about, where they get very good results by combining antibody detection and PCR. So this is the test against the hemolysin co-regulated protein, and you look at the combined data for the two tests, the ICT test and the PCR together, you get a very high sensitivity. The specificity is a little bit low, you think, only 90%, but that's all coming from the ICT test. And they made a note that the ICT was positive in 16 out of 73 culture negative patients. So that was deemed as false positive. So that reduced the specificity to 90. But actually, when they went back to those patients and recultured them, 5 of them became positive. So again, when you're doing evaluations, when you have patients whose diagnosis is hard to be sure of, you can end up misrepresenting the performance of the assay. So the specificity probably is a lot higher than 90, but 90 was their result. So they also make the point that if you take various tests here and you look at different time points in an illness, this is 1 to 3 days of illness, 4 to 6 days, more than 7 days, the tests perform better several days after the onset of symptoms. So in Singapore, what do we do for serology? Well, we don't do it in our laboratory. We send it out to the Defence Medical Environmental Research Institute, and they do an IHA test for us. This is how they define a negative positive. What we always have to remember when we interpret these results, even if we get a positive titer, is we need to know our patient, because if our patient is from another country, it might be that this is just background positive signal. For somebody who hasn't left Singapore, then this is far more relevant. So we need to know our patient before we really interpret serology results. There are, of course, a range of serological assays. The oldest, perhaps most commonly used, is the indirect hemagglutination assay, but it's poorly standardized and uses crude antigens. And of course, in endemic areas, a lot of the population may be positive, so it doesn't really help you in assessing somebody with an acute illness. There's a high background rate of positivity. Interestingly, it's used in Northern Territory in Australia, where melioidosis is very common, as a screening test before immunosuppressing people. I just thought that was interesting. Of course, there are ELISAs against the Palanisamy. And this has been made into an ICT. And when used in combination with an antigen detection by lateral flow, you get pretty good results, but still not brilliant, only 68%. Now there's a rapid version, which has 3 other antigens added into it. And this performs a lot better. Look at the sensitivity, around 90%, and quite a high specificity. So this is that assay, the Melioidosis DS. It's a multiplex dipstick IgG test, including 4 protein antigens. It actually looks like this over here. Here's a negative person, no bands, and here's a positive person where all the bands have come up positive. What's the purpose of me showing you this? This shows each of the 4 proteins, and it shows dilutions along here. Okay, so it's just showing you that some of them dilute out to get higher, are still positive at higher dilutions, and other targets perhaps not quite so good. I also saw this report published in 2024 about CRISPR claiming incredible sensitivity, much better than culture, but I'm not aware that anybody's using this in routine practice. So let's talk about susceptibility testing. Resistance is extremely rare. There are breakpoints from UCAST and CLSI for MICs, but for UCAST, they also give us zone diameter breakpoints. Interestingly, the UCAST people define wild type as susceptible increased exposure for keftaz, cotrimoxazole, doxycycline, and augmentin. And they do this because they want large doses. These patients need large doses. Meropenem and imipenem do have a susceptible category, but these other drugs do not. Now, emergence of resistance actually is very rare. It's usually associated with undrained abscesses. So just to quickly look at the breakpoints, you'll notice that for UCAST, for these drugs I mentioned, they define S as less than 0.001. Basically, they are forcing you not to call these drugs, or to call the organism susceptible to these drugs. They're forcing you, at best, to call it intermediate, which is, of course, between these two concentrations here. I also make the point and show you these pictures that UCAST shows that interpreting cotrimoxazole can be difficult. So they say that the growth within the zone is expected, so you just have a look and draw the zone where you think it probably is, and you can see why this is so subjective and results in people claiming resistance when maybe it's not there. So our data for the last 5 years, we do e-tests on Molahintenagar, we do Paran QC for these drugs here, and we've had 36 patients in the last 5 years. All the isolates were measured, reported as susceptible. I did see variations in MICs, but they all came within the susceptible range. We don't even test gentamicin, so I don't know whether we've had any gentamicin susceptible isolates, if anybody's brought us one back from Sarawak. So I looked back even further and had a quick look at 2012 to 2019. Again, we had a similar number of patients, 48. There was some resistance reported, but I'm not too sure that could have been errors. So let's talk about emergence of resistance. The most interesting thing, of course, is that mobile genetic elements that we see so many plasmids and other bacteria, they're absent from Pseudomallii. So all these things are chromosomally encoded genes. So we see enzymatic inactivation, target deletion, and a lot of efflux pumps. Okay, so it has a pen A-beta-lactamase. We can have deletions in the penicillin binding proteins, and mutations in the efflux pumps, which specifically affect cotrimoxazole. So resistance is still relatively rare. Why? Because we don't catch this organism from other people. So if other people are exposed to these drugs and the organism becomes resistant, the organism is not transmitted to other people, so we don't see resistance in new cases because they're getting it from the soil. So this is just a lovely example. A 60-year-old farmer with septic shock from Laos had Pseudomallii in the blood cultures. They relapsed. The patient relapsed after treatment with meropenem and cotrimoxazole, and subsequent isolates were multi-drug resistant. And so they altered the therapy, and the patient made a good full recovery. And the point here, guys, is that if somebody does relapse, there is value in repeating sampling and repeating the AST, because it might be that they've developed resistance. So let me just show you, these are the isolates they had, 1 to 7. You see that amoxiclavin, ceftazidime remained susceptible, but all these other drugs became resistant. Whole genome sequencing is more and more being used by people in well-resourced places to predict AMR. And these people here, published in 21, they basically assessed Ariba, Card, ResFinder, and AMR Finder Plus, all these softwares, and concluded that they failed to identify any clinically relevant AMR. So they made their own software called RDAP. So I just thought I'd point that out to you. You can look up the details, but it basically performed extremely well, detected all the known changes they wanted it to detect, and accurately predicted 3 previously undescribed determinants. And in mixed strain data, it could identify resistance to as low as 5% allelic frequency. So let's finish off with safety. So you tend to think of this as a hazard group 3 organism, and this old 2004 MMWR is an interesting thing to read. It described 17 lab workers who manipulated the organism outside a safety cabinet. They had 3 weeks of post-exposure prophylaxis, and they all remained serology negative. But the editor made a note that there had only been 2 laboratory-acquired infections previously reported, and one was in somebody who was using open flask sonication, and the other was in somebody who had an ulcerative lesion on their finger when they cleaned up a spill, a laboratory spill. So the editor's notes, they also included another study where 60 lab workers had been followed over 15 years, and 3 of them had titers suggestive of subclinical infection, but these were consistent with the background seroprevalence in the local community. So the editor ended up saying the data suggests that infection is not easily acquired from routine open bench laboratory work. So there's a CMR this year, Clinical Microbiology Reviews, on safety and security in low resource settings for our organism. And they start by saying there's inconsistencies in the safety protocols and risk classifications. It's a rare but severe disease. Human to human transmission basically doesn't happen. But there is, I mean, we do know that it's a low infectious dose, apparently, but still ill-defined. There's no vaccine. Mortality has been reduced to 6%, but still 6% for a lab-acquired infection would still be high. And we end up with it being called RG3 in some countries, but an RG2 in Australia. Of course, the US call it a biothreat agent, and they designate it as a Tier 1 select agent, and the impact of that is the stringent guidelines for possession, storage, use, and transfer. So that's a real pain. So I'd much prefer to work in Australia, where they call it, say, you can use it as BSL-2. So they don't classify it in Australia as a security sensitive agent. And this probably reflects a practical consideration that it's endemic for Pseudomallei, and it commonly occurs in routine laboratories. So I won't read through all of this, but basically I've put down here some references, in case it's interesting for you, of loads of people being exposed to Pseudomallei outside containment, outside safety cabinets. Okay, 30 staff here, handling over 1,200 times, and no infection or seroconversion. An updated MMWR discussing 261 laboratory staff members at risk with a lot of exposure, none developed melioid. Another one here, 51 release events affecting almost 300 people, most of it outside of containment, including spills, needle sticks, and no infections reported. However, this one here, just last year, 160 exposure events affecting almost 900 people. Most of these events were outside containment. 12% did develop positive IHA titers. Okay, but apparently none developed disease. So we end up with David Dance has written a lot about Pseudomallei, and in this paper, he said exposures occur when staff don't realize the identification. So this comes all the way back to how to identify it quickly. And because people don't think about it, they may be a little bit more careless and not use the protective measures they should. But he does say that in endemic areas, such exposures are probably common, but almost never result in infection. It's extremely unusual. So some people, I always say to our staff, organisms don't jump off plates. We grew up sniffing agars, and I think Pseudomallei smells lovely, but I don't do it anymore because I'm told I'm not allowed to. But we did have a look and published some data, as did other people, and they said they couldn't find any error isolation of the organism from doing things like opening the plate, oxidase testing, or preparing them up. And got a one which we hated, so we if you humans there, and they partially open the plate and make a cautious sniff. So we thought the risk of inhaling a significant dose was low. So risk mitigation. Every plate is a hazard, not just Pseudomallei. And when I teach our staff, we always aim at the fact that routine standards should protect against almost everything. And there's a hierarchy of controls. You can't trust people and the PPE, so you should try always to prioritize engineering and administrative measures. So I think I'm almost running out of time, so I'm going to speed up. Focus on using awareness, because if people are aware and question if something could be dangerous, they are going to be more careful. Concentrate on what is really bad. Okay, what is really bad is centrifugation. It's a recognized high-risk activity. So you should try using sealed rotors or safety cups, unloading them and loading them in a safety cabinet and letting them settle before you unload them. You should consider whether your staff may have unseen health issues, as listed here. And staff should have an occupational health review before they work in a microlab. Are we happy with them? Because this CMR document is saying that these people shouldn't be exposed to Pseudomallei on open bench. Because in reality, in most hospitals and most laboratories in Asia, these samples are being dealt with on an open bench. There's a security issue that could be used as a biothreat agent, and they recommend robust physical and cybersecurity measures, but it's very easy to recommend this. It's not so easy to do. The best way to deal with biosecurity is to staff allegiance. Make sure your staff are happy, that they like you, that they like working for their hospital, then they won't do anything bad. So the sustainable practice, I'm finishing off here. I'm basically summarizing everything I've dealt with, that actually the most important thing is avoid the high-risk stuff. So avoid centrifugation unless you handle those buckets in the safety cabinet. And don't send stuff off for washing and reuse without autoclaving it. Okay? So in Malaysia, this is my last slide, and I've now finished. Thank you very much. I would love to know what selective media you use in Sarawak. What tests do you use? Because we get away with moldy, and we also have PCR. So thank you very much.
Thank you very much, Prof, for your enlightening talk on Berkuda Sudamalai. If anyone of the audience have got a burning questions, could you all please raise your hand, or you can type it in the chat box to answer your question.
I can't see the chat box, so
yeah,
if there is a question. I mean, if anybody can answer the questions here for me, I'd love to know about it. What selective media do you use in Sarawak?
Yeah, is there anyone from Sarawak or Sabah in the audience? If anyone is there? Just to get the ball rolling, I've got a few questions, Prof. What is the importance of actually speciating between the Malay, Cepacia, and Pseudomalai, as the clinical manifestation is almost similar? What is the implication in patient management?
Well, the Pseudomalai, of course, causes a far more aggressive disease with sepsis, septic shock, and death. And that can be changed in the mortality brought from some enormous number all the way down to less than 10% if they get drugs like ceftazidime given to them early.
Mm-hmm.
So if we can let people know about that, then that's going to help them in the short term. In the long term, these people relapse if they're not given many weeks, maybe 12 to 20 weeks of eradication therapy. So we don't usually do that for Cepacia. If you think about who we see Cepacia in, we don't tend to see it in people coming in off the street with an acute infection. It tends to people who would be with chronic lung disease, in whom, of course, it can be very harmful.
How about Thailandensis and Malay?
Well, Thailandensis, Malai, we don't really see in people. So we don't want to send reports out saying that these are things which they're not.
Okay.
So I get your point that would it really make a difference?
Mm-hmm.
We won't treat it. So we, I hope they wouldn't. I hope they'd ring us up and say, hey, do you really believe this? But that's why I showed you a little bit on, I know you're asking me to try and generate discussions and you probably know all this. I know you do.
Yeah.
But of course, it's just interesting to read up about the things that we can confuse Pseudomalai with, to understand the different diseases. And actually it makes the whole life so much more interesting if we're talking to our agricultural colleagues and learn about what they see in their animals. And of course, I'm talking about Malai there.
Thank you. I see one question in the chat box by Nurdiana binti Muhammad. Is it safe to transport extracted DNA using routine shipment methods from Pahang to any service provider?
So I always have to be careful about questions because is it safe? Yes, I think it is. If you've extracted the DNA, depending on how you've extracted it, then yes, it would be safe. I mean, one way of extracting is just to boil them up. So we do PCRs on organisms that we just boil them, which kills them and releases some DNA. And then we don't bother using expensive chemicals or kits because there's loads of DNA there. So yes, I think that's perfectly safe. But what I don't know is whether it's allowed. It might be safe, but it may not be allowed. And I don't know what your paperwork is, what you are allowed to do. I
hope that
answers the
question. Yeah, she puts a thumbs up, so she's happy with your answer.
Thank you.
Anyone from AMR from the audience that can answer which selective media is used in Sarawak? I was told that Dr. Rohaida is part of the audience. Are you here, Dr. Rohaida? Anyone from AMR that can answer this question, or anyone from Sarawak? Which selective media?
Do people use selective media at all in Malaysia? I mean, we don't. I'm told that one of the hospitals does have the powder, so they could do, but in our hospital in Tan Tock Seng, we don't.
Okay, there's another question here. Based on my experience, I don't know whether it's a question or it's an answer. The ST881 BP isolates are not completely sensitive, but have reduced susceptibility to both Genta and macrolides.
So I think the point is that selective agents have around 4 milligrams per litre of gentamicin. So I wonder what the MICs are. But I mean, the work is not my work. The reports talk about gentamicin susceptibility. Of course, you could argue they can't be susceptible because there are no breakpoints for gentamicin.
I think this is from Dr. Revati from NIH. This is our National Institute of Health. So she claims that if they prepare the ash down with a lower Genta concentration, it helps identification using the ash down agar.
So basically they use the ash down with I wonder what the concentration is with a lower concentration. Okay, that's interesting.
Yeah.
I mean, when I look at all this data, I think to myself, gosh, I wish I knew the gentamicin results for our own isolates. And I feel slightly foolish that we don't bother testing them because we don't use the drug clinically. But it would nevertheless be interesting to do. There are so many interesting things we should be doing, not just the core work for our patients, but also interesting stuff. Just to know whether any of our isolates are have a lower MIC.
Right. We've got a nice message I think from someone from Sarawak. Sarawak General Hospital uses blood agar and chocolate only.
Do they do throat swabs and rectal swabs? Do you know? Maybe.
Can I just ask? Is this Dr. Valerie? Are you from Sarawak? If you don't mind unmuting and speaking.
Hello, can you hear me?
Yes, you are clear.
Yeah.
Can you just
what's the question again?
Yeah, can you just introduce yourself? I see your chat box there. You've written there Sarawak General Hospital uses BA and chocolate only.
Oh yeah, I'm Valerie. I'm from Sarawak General Hospital. One of the clinical microbiologists here.
Yeah, what was your question again, Prof? Prof's internet is
Gentamicin susceptibility of your isolates.
Sorry, we lost you at the front beginning.
Could you repeat?
Yeah.
Do you use selective agar or selective media for pseudomallei?
No, for our side, Sarawak General Hospital, we don't. We just use like the usual like BA, Chalk, and the MEC conky.
Do you collect throat swabs or rectal swabs from patients you think might have melioidosis?
For now, no, we don't practice that to screen those who suspected with rectal swab. We just receive the clinical specimen for those who are suspected with melio, then they'll send over. I mean, we don't screen them, just like we don't screen rectal swab, yeah.
Do you do any other, I mean, the literature tells us culture is only about 60% sensitive. So a lot of our patients don't have positive culture results. Do you do anything else to try and detect? Do you use serology or immunofluorescence or PCR?
For now, we didn't offer any other tests, serology or IF. We usually, if there is clinicians still suspect, so they will send elsewhere to the referral center like AMR to check for the serology. So for now, we don't offer more than that.
Yeah, we also don't offer anything else, but we send things off to another hospital who can offer a PCR. Thank you.
Sure.
Okay, another comment from Dr. Revati, who comes from the NIH. She says that they distinguish, most of the clinical laboratory microbiologists can distinguish them based on colony morphology. Yeah, they also use blood agar in their case. Dr. Nodiana from Pahang. In Pahang, we get many SXT resistant melioidosis using e-test. How can we validate the results? WGS is helpful, she's asking.
So when you run the e-test, what we do is every time we set it up, we run control organisms in parallel. So, and we have, of course, tolerance limits for what result is expected. So it's quite expensive, of course. In some ways, it's a real nuisance, but we don't see enough cases to do weekly QC. So every time we have a patient with an isolate, we set up the QC. And that gives you more confidence that your result is correct, because I think it's jolly difficult to read the cotrimoxazole. If I were you, I would set that up. If you have set it up and your QC is all working all right and you're still seeing this clinically, and this is in the first sample before the patient's treated, I still don't believe it. because the literature tells me it's so unusual. But who knows, maybe you have something jolly interesting in your environment. I would then go to a reference lab and say, hey, and this is what we do actually, we do this quite often. If we have something odd, we send the organism over to another lab and say, hey, can you just do this for us and see what result you get? So we're benchmarking, we're basically asking for help. We're being very open, saying, we got this funny result, we're sure we're doing something wrong, we don't know what it is. It's going to help us if you can tell us, that yes, we are wrong. So that's how we get better, is by embracing.
I think we have someone who wants to ask another question. She has raised her hand. Can you unmute that, please?
Yeah, Dr. Timothy, I'm a clinician. So, for example, so if I was told about the bacterium resistant, and then that sometimes the patient is actually joint involvement, should I add first bacterium? Do you think because you said just now you didn't, it's actually rare to have a bacterium resistant?
I think the literature is quite clear that it's extremely rare for the isolate to be resistant unless it's been exposed to long-term antibiotics. So, because we acquire these things from the soil, the organisms we get from the soil and the environment haven't been exposed to these antibiotics before in the same way. So it's very different from other organisms that spread from patient to patient, where resistance is spread from patient to patient. This is different.
Sorry, Prof, when you say SXT resistance is rare, so when you run the test, which control that you are referring to that we should include in our experiment?
Acceptable. Yes. Includes ceftazidime. So everybody coming into our hospital with severe community-acquired pneumonia will get ceftazidime because we assume it might be melioid and we assume it will be susceptible. It could be wrong, but it's unlikely. So yes, I would do what you described and assume it's susceptible if the patient hasn't been exposed to it before, that particular drug, the cotrimoxazole.
Okay, another comment from Dr. Muhammad Ridwan, Muhammad Ali from also NIH. He says that in clinical hospital setting, a standard set of culture media is generally used according to the specimen type. For example, for blood culture fluid specimens, we typically use blood agar, McConkey, and chocolate agar. Seborrheic agar may be used for fungal elements if fungal elements are observed or a fungal infection is suspected. Okay, the similar comment also by a student, I suppose, Anis Johari, who has done her internship. Use blood agar and chocolate agar. Again,
These are all okay for sterile samples, aren't they?
yeah. Any other questions? Okay, Dr. Valerie from Sarawak again. She has said that in Kapit, Sarawak, they use the Francis media.
Oh, they do. Okay.
Yeah.
That's interesting.
Yeah.
Thank you.
There's a question for you. In your setting, what solution do you use to prepare the inoculum for antimicrobial susceptibility test for Pseudomalai? Is it tryptone broth or Mueller-Hinton broth or cationic adjusted Mueller-Hinton broth as recommended by CLSI M45?
Well, we follow the recommendation. So actually, right now, without opening the book and checking, I can't remember for each thing which one we use. So I'm sorry, but I'm not doing it day in, day out. But we actually do follow the guidelines, which can be a real nuisance because they can be so different for different things. Do you guys routinely set up controls for your susceptibility testing when you run them? Every time you do susceptibility testing? Is anybody able to answer that? So if you're getting unexpected results, then I think it's very important you do that.
Anyone who's got an answer for that? Your question again, Prof. Anyone who uses control, is it?
Well, I'm just wondering whether you do, when you're doing susceptibility testing, whether you actually set up control organisms at the same time as a way of making sure that your staff are setting up and interpreting the results correctly. Because it can be so difficult reading CVE tests, for instance.
But in practice, shouldn't that be the case, Prof? You should have a control to see the
Well,
value.
they should do, but I'm just wondering. I mean, I'm always open to the fact that people don't always do what we think they do, you see. So I'm always open to ask, what actually happens. Because these things are expensive.
Well, I don't work on Pseudomalai, but of course, for any other organisms that we work on, we do put in the control for validity's sake.
Okay.
Sure, from the rest, anyone who wants to comment about that?
Does anybody use the latex tests or antigen detection tests?
Yeah. Anyone from NIH or Dr. Valerie from Sarawak? Do you use the latex test? Anyone?
I mean, one of the things interesting is that the amount of disease you guys see, your instance data in some parts is very high. And it's just so interesting to see different parts of the world with such different epidemiology. And therefore, maybe you use different
There's an
methods.
answer from Dr. Muhammad Ridwan who says AMR, ORF, TTSS, qPCR. Maybe you want to unmute and say something, Dr. Muhammad Ridwan?
Yeah, in AMR we do qPCR using Novavax 2006, the one that you presented, Dr. Timothy, or
Okay.
number 2 of the sequestration system, and we also offer Meliodosis serology.
So do you do the PCR on the patient sample, or do you do it on the isolate to confirm the isolate?
On the isolates.
Oh, okay.
On the isolates to confirm the ID.
Great.
Meanwhile, detection in Malaysia, it happens at our states or district hospitals.
So do you have MALDI widely available, or is MALDI not so widely available?
MALDI widely available. most of our state hospitals, they have moldy.
So if they have moldy, are they also doing the PCR, or do they just stop with the moldy?
I think this one I need help from our state hospitals to answer. But
Okay.
if I can guess, they don't do PCR for ID of BP.
Yeah, I would guess that too. It's interesting how moldy has changed everything. I mean, I just couldn't believe it when we stopped using gram stains all the time. The new staff aren't used to doing gram stains on all the bacteria because we have moldy. Of course, it's great most of the time.
I think I missed one of the comment from, I believe it's Dr. Amira Amir from MOH Selangor. Something that you commented, need to look for 80% of inhibition as it can be tricky to read.
Yeah.
Yeah.
I look at plates with my colleagues and go, okay, where do you think the 80% mark is? And then we try and clarify in our mind, is it 80% growth or 80% inhibition? Make sure we've got it the right way round. And, and then we peer at it again and again. And it's very easy to come up with different answers. I mean,
maybe I can invite
judge that.
Maybe I can invite her to unmute and speak on your question of the control, whether they have used control. Amira.
Yeah. Hi, Dr. Timothy. I'm Amira here. I'm from Hospital Sungai Buloh in Selangor. So we do a weekly routine QC for Pseudomalai, MIC. And then because we do get the isolates quite often, so we don't wait until we get the isolates. So we do it weekly. And then as for reading the MICs, that really it requires some technical expertise because young MOTs will read it as 100% inhibition, then they will report it as resistant. So we have to look at the colonies again, whether it's truly 80% or not. And
yes.
Yeah, resistance is very rare actually.
Yes, so somebody earlier on said that they felt they saw resistance. So everything you say, I agree with you.
I think
it's
on that, yeah.
yeah. And I think it's so easy to make a mistake.
Yes.
My whole life is spent looking for mistakes, and that's what we do. So we have to be open to them.
If you look at the literature, even in Thailand, they even reported that the initial isolates that were reported as resistant, when they repeat it, it is actually sensitive. So again, there's technical differences between laboratories. So we need to verify that data. And another thing is that with our clinical isolates, most of our samples, we are able to be cultured in the laboratory and we are able to identify with Melioidosis. But then some patients who we could not get any positive cultures, then we can ask our AMR colleagues to do the Pseudomalai PCR.
Great, so you have backup.
Yeah. Yes.
Yeah, so we have backup as well. We don't do the PCR here, but we can send it to another lab to do it.
Yeah.
I mean, we could set it up, but there's no point. Singapore's so small. It's not.
Yeah. All right, thank you.
Thank you. One last question, Prof, from Dr. Muhammad Ridwan again. Are you with us or have I lost you?
Yes.
Okay. In Singapore, do you handle BP in BSL3 or BSL2?
Okay, well, routinely every day we work at BSL2. And I think this is what people understand, is to be realistic. We could grow it from any sample. So it could turn up on any bench, it could be on the urine bench. It could be on any bench. And we're not going to put the whole lab in a BSL3. So by the time our staff see something on a plate, they've opened it in a BSL2 in the open air. It's not in the safety cabinet. If they recognize the colony, or they think, gosh, this could be a Pseudomalii, then our operating procedure is that they then handle it inside a biosafety cabinet. And then they sellotape the lid on so people can't accidentally lift the lid without thinking, oh, why has this got sellotape on it? So we do that for anything where we think there may be something dangerous. So once we realize that, or have a suspicion it goes into a biosafety cabinet is the answer.
Okay, I think with that, I would stop the question and answer session. Thank you very much to all the participants, and thank you very much to Prof. Timothy to actually to come online at this time at your busy hour. Till we meet
Thank
again.
you.
Yeah, till we meet again. Thank you very much to everyone. For
Thanks
those
very much.
participants, you can submit your attendance and claim your CPD points. Please check the chat box. Thank you very much, Prof.
Thank you.
Have a good day.
Bye-bye.